DNA fingerprinting. The chemical structure of everyone's DNA is the same. The only difference between people or any animal is the order of the base pairs. There are so many millions of base pairs in each person's DNA that every person has a different sequence. Using these sequences, every person could be identified solely by the sequence of their base pairs. However, there are so many millions of base pairs, due to which the task would be very time-consuming. Instead, scientists are able to use a shorter method because of repeating base patterns in DNA, satellite DNA. These patterns do not, however, give an individual fingerprint, but they are able to determine whether two DNA samples are from the same person, related people, or non-related individuals. The use of DNA probes and the development of retrieval techniques have made it possible to match DNA molecules to one another for identification purposes. This process has been used in a forensic procedure called DNA fingerprinting. DNA fingerprinting depends on the presence of RFLPs, the repeating base sequences that exist in the human genome. As the pattern of RFLPs is unique for each individual, it can be used as a molecular fingerprint. 1. The DNA is extracted from the nuclei of white blood cells or of spermatozoa or of the hair follicle cells that cling to the roots of hairs that have fallen or been pulled out. 2. The DNA molecules are first broken with the help of enzyme restriction endonuclease called chemical knife that cuts them into fragments. 3. The fragments are separated according to size by gel electrophoresis. 4. Fragments of a particular size having VNTRs, variable number of tandem repeats, are multiplied through PCR technique. They are treated with alkaline chemicals to split them into single-stranded DNAs. 5. The separated fragments of single-stranded DNA are transferred onto a nylon membrane. 6. Radioactive DNA probes having repeated base sequences complementary to possible VNTRs are poured over the nylon membrane. The method of hybridization of DNA with probes is called Southern blotting, after the name of the inventor E.M. Southern, 1975. The nylon membrane is washed to remove extra probes. 7. An X-ray film is exposed to the nylon membrane to mark the places where the radioactive DNA probes have bound to the DNA fragments. These places are marked as dark bands when X-ray film is developed. 8. The dark bands on X-ray film represent the DNA fingerprints is equal to DNA profiles.